Yes. તો તમારું મટીરિયલ આપી દઉં તમને.
ચેક મોકલાવો જીજેલા સર એમ કીધું કે આરટીઆઈ અને સ્પીડ પોસ્ટ બંધ છે તમે જે તે પાર્ટી ને તે નંબર માંગીને ચેક જાતે જમા કરાવી શકો.
કરાવી દે હું બધા પાસેથી લઈ લઉં છું.
હા તો મારી પાસે ના જમા.
So yesterday we left with QTL analysis. We'll again continue that. So this slide deals with statistical aspect of QTL analysis. So once we prepare the molecular markers from our mapping population, we have the molecular marker data also. MapMaker program will give you the molecular marker data. And then this marker data will be associated with the quantitative trait. Yesterday, I also told you that by grouping of different RILs on the basis of marker genotype and calculating the mean values of those group for the trait, if those mean values are significantly different from each other, we conclude that marker is linked with the QTL. And if they are non-significant, we said that marker is not linked. So this is the way we identify the marker linked with the particular QTL. Now this session deals with identification of significance of that identified relationship. So how we can say the relationship we have identified between marker and QTL is significant, and how we can measure the effect of each QTL to a particular quantitative trait. So for that, regression association is utilized. T-test is of course utilized to test the significant difference between mean. If we consider the maximum likelihood method, then load score and R-square values are utilized. So there are basically two aspects considering the statistics used in QTL analysis: regression and maximum likelihood method. Now load score, I already told you yesterday, it is ratio of linkage to no linkage. No linkage means linkage by chance. So sometimes due to statistical errors, I mean the errors in your experiment, or due to chance factor, we may get the presence of linkage between marker and QTL locus, but it may be due to chance factor. It is not the real linkage. So to distinguish this real linkage from chance linkage, the load score is utilized. Higher the value of load score, it indicates significance of QTL. So if load score of 3 is generally preferred, sometimes 2.5 is also good. Nowadays we'll find a load score value more than 6 or 7, because now we have good molecular marker techniques, SNP based genotyping methods, by which we can identify many polymorphisms across the genome. So this is the load score threshold value, and as I told you yesterday that we get the peak, and if the peak at this peak are indicating the association between a particular marker locus and the trait value, phenotypic value of the trait. And And we need to fix this threshold value. Threshold value of 3 indicates that means out of 1,000 cases, the error probability is 1. It is 1 out of 1,000. In 1 out of 1,000 cases, the indicated linkage is due to chance factor. 999 times the linkage is real. That's the error probability. So with that probability, if the peak is going up beyond this line, threshold value of load score 3, then we call it significant QTL. So there are many ways we can associate marker data with the trait value. There are single locus analysis method and interval mapping method. So let us consider single locus analysis first. Single marker analysis can be implemented as it can be utilized where our assumption is a single QTL and a single marker is linked with the single QTL. We can handle a single QTL locus as we are handling any qualitative locus. A locus is responsible for any qualitative trait. Similarly, the QTL single locus is also handled like that. And here we use ANOVA. We can analyze the phenotypic variance of the trait under consideration. And we can also classify the marker effects. Marker effects means how marker is contributing to the trait. Generally, to establish the association and regression between this, linear regression is utilized. And we can also use load score maximum likelihood method. These are the advantages of single locus analysis. It is very simple. In terms of data analysis and implementation, we don't need dense linkage maps and gene orders for this particular kind of analysis. But there is a disadvantage of this method. We will find a confounding effect. So if you have not studied confounding in statistics, confounding can be understood by a simple example. There is a third factor which interfere with the correlation or association between the two variables under consideration. So we can understand that with one example. There was one survey in America that the survey dealt with the number of people eating ice creams and the number of people who are drowned in swimming pool when they go for the swimming. How many people are killed because of drowning? And there was a correlation. There was perfect correlation for the people eating ice creams and people killed by drowning in swimming pool. So we know very well that that's not possible. Eating ice cream and drowning in swimming pool, they are not related to each other. There is no biological meaning to that. But then it was found out that this observation was taken during summer season. And we know that during summer season, people eat more ice cream and more people go to swimming pool. So there was a third factor, a season, summer, that was confounding this result. And because of that summer season, the nonsense correlation was observed between eating ice cream and people drown in the swimming pool. So that's the confounding. So here we have the same problem of confounding effect. What is confounding effect here in this case? See, we have one marker and we are associating that marker with quantitative trait. Quantitative trait is measured phenotypically and is the final outcome of all the alleles present in that particular individual. When you observe, let us say, pods per plant or any other trait from the mapping population individual, that individual, when you measure number of pods per plant, it's the final outcome of all the effects of the alleles present within that plant, positive, negative, interaction between them, linkage between them, so many things are there. But when we are measuring a particular marker, we are limiting for marker locus, it is located at one locus only. And when we measure the trait, it's a final outcome of all the alleles present in that individual. So that's the reason for confounding effect. QTL interaction, linked QTLs, we are measuring the phenotype, the final outcome. And because of this confounding effect, QTL effects and position cannot be measured precisely. I mean, if you identify 10 markers, let us say, linked with various loci governing a particular quantitative trait. And the effect of each marker cannot be precisely estimated in the single marker analysis or single locus analysis because of the confounding effect. And positions cannot also be estimated properly. You may find many markers associated with the QTL, and you cannot say which marker is more significantly associated with the QTL. Because in single marker analysis, generally gene order maps are not available or if they are not utilized. And because of that, there are few markers studied. And this technique was proposed initially before other sophisticated techniques were found out for QTL analysis. So next best technique, improvement over single locus analysis is interval mapping. Interval mapping, we consider flanking markers, and between these two markers, whichever interval is present, that marker interval may be harboring one QTL, one locus governing quantitative trait. So joint frequency of these flanking markers are utilized. These markers are flanking the QTL locus, a probable QTL locus. Let us say in single marker analysis, we use one marker which is linked with the QTL, and here interval interval mapping, we utilize two flanking markers which may be harboring a probable QTL. So that's called interval mapping. In interval mapping, we get more precise estimation of association of this locus with the QTL if it is there, if it is present, contributing to that quantitative trait, because two markers are considered at a time rather than the single marker. So there is advantage of considering two markers, flanking markers, as compared to single marker. And that could be understood by this particular slide. Now here, there is one QTL. And suppose we identify a marker which is 5 centimorgan away from this QTL. In decimal terms, it is 0.05. 5% recombination, that means 5 centimorgan. It means there is a 5% recombination between this marker locus and QTL locus. Now, if we use marker A only for selection of that QTL, or to identify that QTL, the selection efficiency is 95%. That can be calculated as 1 minus recombination between marker A and QTL. So 1 minus 0.05 is 0.95, it's roughly 95%. It means that if you identify this marker A, and if you utilize this marker for selection of that QTL, if you genotype 100 individual, and in all these 100 individuals, this marker A is present, then you are sure that in 95 out of that 100 individual, QTL is present along with the marker. In remaining 5 individual, marker A might be present, but there is no guarantee that QTL is also present. That is the meaning of 5% level of significance statistically. Now, if we utilize two markers, flanking markers, marker A here and marker B on another side, the distance remains the same, 5 centimeters. So if we use marker A and B together to identify that QTL or select that QTL, the efficiency is 1 minus 2 multiplied by recombination between A and QTL and recombination between marker B and QTL. So that would be roughly 99.5%. 1 minus 2, recombination between marker A and QTL is 0.05. Here also it is 0.05. So if you calculate it, it is 99.5%. It means that if you have selected 200 individuals carrying both the markers, then in 199 individual, you are sure that particular QTL is present. That's the efficiency of utilizing two markers. So that efficiency is too high. So this is the advantage of utilizing flanking marker over single marker. So that's the interval mapping. We utilize two flanking marker and the marker interval, and we try to test whether a particular marker interval is significantly associated with the trait or not. When you look at the papers on QTL, you find these kind of the graph. For simple understanding, I have kept only one chromosome here. You get the marker map data. This is the centromere. You get all this marker map data when you perform molecular marker mapping. And then you associate this marker maps with the particular quantitative trait. This dotted line indicates a load value of 3. And you can see here, this peak is going beyond this dotted line, and it shows the particular marker shows the value of 7. So it indicates that it's significantly contributing to the trait. This is the same explanation for load scores. I'm not going to repeat. There are also limitations with the simple interval mapping. The number of QTL cannot be resolved because sometimes 4 to 5 QTLs may be linked together. And if you utilize a flanking marker to this QTL, 4 to 5 linked QTL, QTL analysis will tell you that there is a single QTL present within, but actually there are 4 to 5 QTLs present. So in case of linked QTL, simple interval mapping cannot give you better result, because it will tell you that single QTL is present at that, between these two flanking markers. Actually there are 4 to 5. So we cannot estimate number of loci governing a quantitative trait properly, precisely. The location of QTL is also not resolved. An exact position cannot be resolved because this marker interval may be still too longer to precisely locate the QTL in the narrow region. So if you use dense marker within a limited region, then it would be possible. But here we are only considering 2 flanking markers situated at the boundaries of this particular interval. And statistical power is still low with the simple interval mapping. And all these limitations are due to these reasons. QTLs might be linked together. They cannot be resolved properly with simple interval mapping. There might be interaction between this QTL, and that's the reason also the position cannot be properly estimated. And limited information in the model. The model which is utilized for simple interval mapping and QTL analysis, and it doesn't have the capacity to estimate epistatic interaction and gene interactions properly. So these are the problems with simple interval mapping. Now the major problem with simple interval mapping is linked QTL. So to resolve that aspect, composite interval mapping was proposed. So composite interval mapping combines both simple interval mapping and simple interval mapping and single locus analysis by utilizing multiple linear regression to find out the markers associated with the QTL. So it is based on simple interval mapping and single locus analysis. So it will utilize 2 flanking markers, first of all, and try to know whether this region is associated with the quantitative trait or not. Apart from this 2 flanking marker, this is our QTL and linked QTL, might be 4 to 5 QTLs linked. Apart from this marker interval, some single markers are utilized, situated within this region. Now, if there is single QTL, then all this marker will show the same effect on quantitative trait because there is single QTL. If there are many QTLs here, then in this individual marker will show different effect towards the trait. So if different effects are different, estimated by the multiple linear regression, then there are linked QTL present. So this is another question for competitive exams. Composite interval mapping is utilized to resolve the linked QTLs. So QTLs are linked. If you want to resolve whether there is a single QTL or many QTLs linked together, then we utilize composite interval mapping. Multiple interval mapping. So now rather than utilizing the single markers, we utilize the intervals. Within this region, we utilize the intervals and we try to map the intervals with the quantitative effect and effect on the trait, and we utilize the multiple intervals to find out whether these QTLs are, if there's a single QTL or these QTLs are linked or not. So these are the major methods for QTL analysis: single locus analysis, then interval mapping. Under interval mapping, we have simple interval mapping, composite interval mapping, and multiple interval mapping. These are the softwares utilized for QTL mapping. I'm not going to describe them, it's not necessary, but most widely MapMaker QTL is utilized for QTL analysis to construct the marker map data and QTL analysis. A summary of the statistical methods used in QTL analysis. Statistical methods uses F-test to know the significance of the variance, trait variance. Uh, the variation present in the trait is partitioned into various marker effects. Utilizing the regression approaches also, the marker effects are estimated and results are generally presented in the tabular form as we normally present the results of any ANOVA. Likelihood approach utilizes logarithm of odd, real linkage to chance linkage ratio to test the statistical significance of particular position. That's how load score is utilized. So in older QTL papers, if you refer around 2005 to 10, generally this load score of 3 is considered better. But nowadays, to decide the load score for a particular experiment, what a statistical solution is utilized. In that case, suppose you have the markers, you have the quantitative trait, and you have identified several significant QTLs. Now, the data are shuffled now. Either the marker effects are kept constant. I mean, the molecular map is kept constant, and the data of RIL is randomly shuffled. So this random shuffling results in chance factor, and by random shuffling, again the QTL analysis is performed. With that random shuffling, also if you are getting significant QTL, then it indicates Hall's discovery ratio. It indicates linkage by chance because now you have randomly reshuffled the trait value, RIL trait value. You should not get the linkage when you randomly shuffle the trait value, keeping markers constant. But you are still getting, that indicates the false discovery ratio or chance factor. So that is again taken into consideration, and accordingly the minimum load score is decided. In some of the experiment, minimum load score may be higher than 3, 5 or 6 depending upon the shuffling and discovery of, measuring the false discovery ratio. So that is generally followed nowadays. I mentioned all these things. In backcross, dominant and codominant marker will perform equally. In F2 progeny, you can utilize dominant marker, but the information you will get is less reliable as compared to codominant marker. Regression approach for simple interval mapping and composite interval mapping is easy to implement, and it gives the same result as the likelihood approach. Generally, most of the paper you will see both the approaches have been utilized. Likelihood approach to find out the significant QTLs, and the regression approach to work out the marker effects in terms of percentage phenotypic variation expressed. So to know that, you have to go through the papers on QTL, then you will realize what is this effect. And with this QTL analysis, nowadays statistical methods are available by which you can also measure additive dominance and epistasis utilizing markers in QTL analysis. So that is also possible. Composite interval mapping, as I told you, it has benefits over simple interval mapping because it can resolve the linked QTLs. Now, factors affecting power of QTL mapping. So what factors are responsible? If you want to map QTLs precisely, then these are the considerations. These factors affect your QTL analysis, power of QTL mapping. Number of genes controlling the trait. So more the number of genes controlling the trait, the power of QTL analysis will be low. If there are few QTLs controlling the trait, you will be able to map the loci precisely. But if there are more loci, then you have to work hard. You need more dense marker map, you need more number of individuals, mapping population individuals, and their genomic positions. As I told you yesterday, if these QTLs are linked, then it will decrease the power. So you have to go for composite interval mapping. If some of the QTL is very much close to centromere, then also it will be problematic to work out the linkage distances between marker and QTL. Distribution of genetic effects and gene interaction. Genetic effects mean whether, when suppose 10 loci are governing the quantitative trait, there might be additivity between 4 to 5 loci. For other loci, there might be dominance. 2 to 3 loci might have the epistatic interaction also. So how these gene effects are distributed across the loci governing a particular quantitative trait, that is also important, because at marker level everything is qualitative. Even the single gene actually contributing to the quantitative trait is Mendelian. That means it will have one gene and two alleles in normal diploid population. But all the complexities are arising additive, dominance, epistatic at phenotypic level, when the protein products of this loci are interacting to yield finally the final outcome, which is you are measuring as morphology. I mean, you are measuring morphologically. Heritability of the character. Before going to the heritability, more the genetic interaction, more the epistatic interaction between the loci, the power will be lower, because most of the methods even nowadays available, they cannot take into account all the epistatic interactions. Heritability of the character. Higher the heritability, there will be precise estimation of QTLs. Because high the heritability, environmental influence is lower, and most of the phenotypic variance present in the population, it is expressed by the genetic means or the QTLs, DNA fragments. But if the heritability is low, it means if there is more environmental contribution to the trait, then the power will go down. Number of genes segregating in the segregating population. It is very interesting to understand this aspect. Suppose there are 10 loci governing the trait. So you'll be crossing two parents which are diverse for that phenotypic trait. But actually in that, considering these two parents, out of 10 loci, 8 loci might be diverse. Means each parent is having diverse allele for these 8 loci. But for 2 loci, these parents are having identical alleles. That may happen. And when that happens, actually 10 loci are contributing to the trait, but in your mapping population developed from these two parents, only 8 loci will be segregating. And the markers will always focus on the polymorphism. And even if with these identical alleles present in the mapping population, two alleles are identical, two loci having identical alleles, even if there is a polymorphic marker linked with that, I mean those loci, those two loci, you will not find linkage between this polymorphic marker and the loci, because those two loci, QTLs, are not segregating in the population. So it is also important how many genes are segregating in the mapping population, and that's why the parents we select must be as diverse as possible. If they are genetically related, then the power will be low. Mapping population type and size. So as I told you, RILs are most widely utilized because of higher number of recombination events. And the size. Minimum 200 individuals should be there to take into account all the recombination frequencies and all the markers. If you use the dense marker map, then it is better you increase the mapping population size up to 300, 350 or 400. It's better if you can have 400 individuals. But definitely with 400 individuals and more markers to be genotyped, you need more resources and more fun. But the minimum criteria is 200, and for the marker, you can try with minimum 50 markers. But if you look at the papers, good journals nowadays, papers of 2019 and 20, then 400 individuals have been genotyped with so many markers. There is no limitation over number of markers could be utilized due to availability of this genotyping by sequencing technique to genotype many individuals at a time with many markers. Statistical methodology use, whether you use regression, whether you use maximum likelihood method, whether you go for simple interval mapping, composite interval mapping, so that's decides the power. And significant level use for QTL mapping. So generally, as I told you, that load score of 3 is considered better, but by shuffling method, as I described earlier, if you realize that false discovery ratio is higher in your material, then it is better to raise the significance level from load score of 3 to 4 or 5, where you don't get the false and you don't get the linkage due to chance factor. And the marker density. More the marker density, it is better. If marker density is too low, then the power of QTL mapping will go down. So marker density should be higher, and along with the density, the marker must be distributed randomly across the genome. Only density will not. Somebody may say that I have utilized 1,000 markers or 500 markers for QTL mapping, the polymorphic markers, 500 polymorphic markers. But if these markers are only distributed across 2 to 3 chromosomes, then power will go down. So it is advisable to use 500 polymorphic loci which is distributed across the genome rather than 500 loci limited to 2 or 3 chromosomes. It's better if you utilize 500 polymorphism distributed across the genome. Chances of getting a link marker increases with this randomly distributed dense marker map. Limitations of QTL mapping. QTL are hypothetical genes estimated or identified on the basis of statistical inferences. So in a particular experiment, out of 10 loci you have identified, one may be a false QTL because of many reasons. QTL interaction, link QTL, so many reasons. So few QTLs may have little biological meaning. So it has to be worked out with experimentation. Once the QTLs have been identified, they are to be validated in a diverse other germplasm population or diverse population in another mapping population. Suppose you have worked out 10 loci governing a quantitative trait from one population, it is better to validate those loci in other mapping population developed from other parents. Or you can validate that in germplasm lines, or you can validate that QTL. Suppose you have identified QTLs from F2 population. It is better to validate those QTL in F3 and F4 in the same population. Genetic models for QTL mapping are not accurate because, as I told you, that most of the models do not incorporate the epistatic interactions. Mostly they are present, these quantitative traits. Statistical methodologies are not yet powerful. So these are the limitations, but still successfully loci may be mapped, loci can be mapped, which are linked with particular quantitative trait or associated with particular trait. So QTL analysis is also one kind of association analysis. The concept of linkage is utilized to construct the molecular marker map. Concept of linkage is utilized to identify a marker which is linked with QTL, but when you decide the linkage between marker and QTL on the basis of the significant mean and other concepts like association, you are actually associating intervals with the quantitative traits. So linkage is utilized to construct the molecular maps, and then we associate markers and intervals with the quantitative trait. So it is also one kind of association analysis. Association and regression is utilized to integrate the marker information, marker map information, and the quantitative trait so that we can identify ultimately the markers which are linked with the QTL. So that was all about QTL analysis. So I will wait for 2 to 3 minutes if there is any question related to QTL analysis. Otherwise, I will continue with association mapping and linkage disequilibrium. So if you want to attend the lecture, do not leave because I am going to continue up to 11 and 12:15. I am going to cover linkage disequilibrium, the basic concept of linkage disequilibrium today. If you have any question, then we can wait. I'm not going to finish the lecture. Okay, then we can handle the questions at the end. So let us start with association mapping. So for gene mapping, that means identifying the marker linked with the genes or mapping of the genes on chromosome and across the genome. There are two basic strategies. One is linkage mapping, another one is association mapping. Linkage mapping is based on concept of linkage. And association mapping utilizes linkage disequilibrium. Linkage and linkage disequilibrium are two different things. We will know how they differ. So linkage disequilibrium, first time it was defined by Jennings in 1917, and then it was quantified. The formulae were given in 1964 by Newton. And first time association mapping was applied in plants by Thornsbury et al. In 2001 in maize. So we started applying association mapping in plants since 2001. And then there were many improvements, there are many methods. Association mapping was limited to animals. In plants, scientists were not utilizing association mapping. Because in plants, mapping populations are possible. We can develop mapping population by selfing, RILs, double haploid, AILs, all these populations can be developed. But in humans and animals, these mapping populations cannot be developed. So in the first time association mapping, I think it is more popular for human genetics and animal genetics because mapping population could not be developed. In plants, it was easy. But then later on, the importance of linkage disequilibrium and association mapping was realized. And scientists started applying association mapping in plants also. So first time kind of association as a mapping tool. Association means correlation. As a mapping tool was first time described by Carl Sachs in 1923, where he morphologically associated size differences in the seeds with seed coat pattern in beans. Phaseolus vulgaris, the crow. So first time it was utilized, association as a mapping tool. Now linkage mapping has certain disadvantages. First of all, linkage mapping is very costly because you have to develop mapping population. It's time consuming and needs more resources. And any biparental population we utilize for linkage mapping, it evaluates only 2 alleles per particular locus. In nature, it is not the case. For most of the loci, there are multiple alleles present in nature. Linkage mapping only evaluates 2 alleles at a time in any biparental population. And linkage mapping considers real-time recombination. Real-time recombination means recombination which took place during the development of that population. Like for double haploid, recombination occurs in F1 meiosis only. We take advantage of that recombination only, where the chance for recombination is given only once in F1 meiosis. Because of this low recombination event, even in RILs also, recombination events are still low. And that's why we don't get much finer resolution. We don't get the markers of polymorphism which are tightly linked or more close to the gene of interest. We get loose markers, loosely linked markers. So resolution is still low. I will talk about that. I have a slide. I have illustration by which we can understand this. So QTL analysis identified very few loci governing the quantitative trait. That's the reason. So association mapping is a population-based survey. It identifies the trait marker association or trait marker relationship based on linkage disequilibrium. Linkage disequilibrium exploits historical and evolutionary recombination events. So by linkage disequilibrium, LD mapping and association mapping, we can also take into consideration the recombination events that happen in the history of that crop, the evolution of that crop. We can do that. And in association mapping, germplasm panels or germplasm lines can also be subjected to association mapping. So mapping population need not to be developed for association mapping. You don't need mapping population to perform association mapping. So these are the advantages of association mapping. Mapping population is not to be developed, and we get fine resolution due to consideration of many recombination events took place in the history of that crop, evolutionary history of that crop. These are the advantages. We consider germplasm lines and more alleles, not only 2 alleles, so that broader genetic base gives us better idea about marker trait correlations and associations. Many alleles are evaluated simultaneously. When you genotype a germplasm panel, let us say 200 germplasm lines for a particular polymorphism, at single marker locus, you will get many alleles, multiple alleles. At single locus in diploid organism, there will be 2 alleles, but at population level, there might be more than 2 alleles, like our blood group, like self-incompatible. So many alleles are evaluated simultaneously in association mapping. So that gives better resolution, better understanding of trait marker relationship. The mapping resolution is high, as I told you. We consider many recombination events. We can also exploit historically measured phenotypic data. Suppose you have one germplasm panel, and in 2004 or 2005 or 2015, you measured a particular trait, and you have maintained those germplasm lines. You can genotype those germplasm lines, and you can associate those marker data with the historically measured phenotypic values, trait values. That is also possible with association mapping. And no need to develop the mapping population. So that reduces the cost and that saves time. But nowadays, by applying speed breeding, we can also develop RILs within 1 year in most of the crops. So we have to compare linkage mapping and association mapping with that sense. So before understanding association mapping, we must be clear what is linkage disequilibrium. Simple definition and most widely used definition for LD is a shorter form of linkage disequilibrium. LD is non-random association of the alleles at various loci. Random association means association due to chance factor. There might not be real association, but random association. Non-random association means statistical association. Association between 2 alleles of 2 loci. See, when I say alleles, we are not talking about single locus and 2 loci. We are talking about various loci. So non-random association between the alleles of 2 loci is linkage disequilibrium. We can measure the LD, we can create LD map between many loci situated across the genome. So it describes the non-equal haplotype frequency. What is haplotype? In haplotype, we consider single chromosome or group of the alleles received from a single parent. That is haplotype. It is opposite to genotype. Genotype, you can also say genotype means zygotype. So if you know the difference between haploid and zygote, gamete and zygote, then you can infer, then you can understand what is haplotype. So we consider haplotype frequencies, single chromosome at a time. Allelic constitution or composition of single chromosome at a time received from particular parent, that is haplotype. So by definition, it is non-random association. So what it means? So let us say capital and capital B are 2 alleles of 2 different loci. That means A is one allele of locus A and B is another allele of locus B. There are two loci under consideration. So haplotype frequency where this A and B both are together in one individual, together, co-occurrence of these two alleles. If this co-occurrence is not is equal to the multiplication of their independent frequency, then we say that linkage disequilibrium is present. How we can understand this? So it is again, this formula is based on very similar to the probability rule for independent events. When we say that there are two events, if they are independent from each other, we multiply their individual probability. So if two events are independent from each other, that means if they are not associated. Here two events are presence of A in the individual and presence of B in the same individual. So that is the joint occurrence. Now, if these two alleles are independent from each other, if they are not associated, then their joint occurrence will be equal to multiplication of their independent occurrence. That means they are independent, they are not associated. In that case, their joint probability will be equal to the multiplication of their independent probabilities. But if there is some kind of non-random real association between these two alleles, then their joint occurrence will not be equal to multiplication of their independent probability, but their joint occurrence would be much more higher than multiplication of their independent probability. So we can derive the formula for linkage disequilibrium. D is equal to the joint occurrence of any two alleles of any two loci minus multiplication of the independent occurrence. So that gives us one quantity, one estimate, which is called D. So you see here, if these two alleles are independent from each other, then their joint occurrence would be equal to the multiplication of their independent occurrence. So if these two terms are equal, then D is 0. When the D is 0, it is called linkage equilibrium. There is no disequilibrium. And even if you find association between these two alleles, and when D is equal to 0, it is random. It is due to chance factor. When D is not is equal to 0, when you get some estimate here, not 0, then we can say there is a linkage disequilibrium between these two loci. So significant LD can occur between alleles at different loci, even if they are present on different chromosomes. For linkage to occur, two alleles, two loci must be physically close together on the chromosome. But for linkage disequilibrium, it is not necessary. Two loci may be physically linked close to each other, may they show linkage. Then LD will also be higher, because if two loci are linked, they are always going to be associated together from generation to generation. So higher the linkage between two loci, we will get higher LD also between these two loci. But when LD is high, when there is a linkage disequilibrium, when these two alleles or loci are associated with each other, it doesn't mean that they are linked, but still they are associated. That's the difference between linkage and linkage disequilibrium. So linkage disequilibrium can occur between the loci which are there on separate chromosome because of the factors other than recombination. There are other factors also: mutation, migration, selection, genetic drift, genetic diversity. There are many factors. We'll consider those factors later on. So to understand this LD, how we calculate LD, I have one example where A1 and A2 are the alleles of one locus. These are the SNP alleles. A1 means thymine is present. A2 means at that place in A2 allele of this locus, cytosine is present. We have another locus B1 and B2 are the SNP alleles of that locus. So let us consider haplotype frequency judged by any method to detect the polymorphism. Let us say by GBS approach. By genotyping, by sequencing, we could know the allelic status of all these haplotypes present in a population. These are the symbols for their haplotype frequency. X11 means the individual haplotype having A1 and B1 allele together. A1 and B2, A2 B1, A2 B2. This is the haplotype frequency, 0.6, 0.1. Remember, the total would be 1 because we are talking about allelic constitution at a time. So it is more related with the population genetics. Now we can work out the allelic frequency from this haplotypic frequency. Let us give symbol P1 to A1, frequency of A1, P2 to frequency of A2, Q1 to B2, Q2 to B2. These are the allelic frequency, these are the haplotypic frequency. Now allelic frequency P1 of A1 would be 0.7. That can be judged from this. How? Wherever A1 allele is present for the haplotype, we just sum up the haplotypic frequency. 0.6, A1 is present here, 0.1. 0.6 plus 0.1, 0.7. Because these are the haplotypic frequencies, so we can directly sum them. When we say haplotypic frequency is A1, B1, in this 60% individual haplotypes, out of total haplotypes under consideration, in 60%, A1 allele is present. That's the idea. In 10%, A1 is also present, but here in this 60%, A1 is with B1. In this remaining 10%, A1 is with B2. So 70%, you can say, or 0.7 is the frequency of A1 allele. Frequency of A2 allele is 0.2 plus 0.1 because in this haplotype A2 is present, so it is 0.3. For B1, 0.6 plus 0.2 because B1 is present in these two haplotypes. It is 0.8, B2, 0.1 plus 0.1, 0.2. So now if you sum up these two frequencies of locus A, P plus Q is equal to 1, remember. Here P1 and P2 are the frequencies of these two alleles of a locus. So P1 plus P2 is equal to 1 and Q1 plus Q2 is equal to 1. Now before going to calculate D and other parameters which are used for LD mapping, let us understand some other aspect of D. So value of the linkage disequilibrium will vary from minus 0.25 to 0.25. And based on the concept of linkage disequilibrium LD is influenced by the allelic frequencies. So we need to normalize the value of D before incorporating that into analysis. It is called normalized D. D dash is the normalized D value. How it is influenced by the allelic frequencies. So we are considering two loci at a time here, locus A and locus B, according to the previous slide. Now when you pool germplasm panels for association mapping, the allelic frequencies might be different in different germplasm lines. And those unequal frequencies present may bias the estimation of LD. That's why we normalize the value of D. The rho D value is normalized across the allelic frequency. So here, if the D value is greater than 0, then we have the measure of D dash, which is called D max, is equal to D, the value of linkage disequilibrium divided by D max. D max is the smaller of these two. So you have to multiply these frequencies here. P1, P2, Q1, Q2 is there. So you have to multiply these frequencies and we put smallest of these two as denominator. So that would be normalized D value. If D value is less than 0, then normalized D value is D divided by D minimum. And D minimum, this is actually D minimum, the normalized D dash value. D minimum is the larger of these two. So you see, to calculate the D maximum, we are dividing D by smaller value. And to calculate D minimum, normalized D minimum, we are dividing this D by larger of these two. You can see the combinations are different. So this is how D value is normalized to normalize the D value across the allelic frequencies. Now there is another measure, R square, which is also related to, now the value of D is analogous to the correlation or association, and R square is analogous to coefficient of determination. So R square is equal to D square divided by multiplication of frequencies of all four alleles under consideration when you consider two loci at a time. If R square is 0, then these loci are in complete linkage equilibrium. There is no linkage disequilibrium. When R square value is 1, there is perfect linkage disequilibrium. So R square value ranges from 0 to 1, and the D value ranges from -2.5 to +2.5. Sorry, -0.25 to +0.25. We'll know in the next coming slide what is the importance of R square and D. So let us first understand how we can calculate D value. So I already explained how we can calculate allelic frequencies from haplotypic frequency. Now we calculate D. So the joint occurrence, here we put the frequency of A1, B1 haplotype, because in those haplotypes, A1 and B1 are together. So it is 0.6 minus multiplication of the independent frequencies minus frequency P1 is 0.7 and Q1 is 0.8. 0.7 multiplied by 0.8 is 0.56. So 0.60 minus 0.56 is equal to 0.04. So this is the rho D value calculated this way. We can calculate D many ways from the same data. And there is another formula for D, X11, X22, haplotypic frequency where A1, B1 are associated and A2, B2 are associated, minus haplotypic frequency of A1, B2, A2, B1. That will also give us the same value, 0.04. We can also calculate other way. Let us consider D value from X22. So D is equal to X22 minus frequency of A2 and frequency of B2. That means P2 and Q2. So X22 here is 0.10. Remember this, 0.10 minus P2 multiplied by Q2, 0.3 multiplied by 0.2, it is 0.06. So 0.10 minus 0.06 will also give you 0.04. So this is how we can calculate the rho D value. This is how it is calculated. Now D value is greater than 0, is 0.04. So now we'll calculate the normalized D value, which is D divided by D max. And D max is smaller of these two. So we multiply P1 and Q2. 0.7 multiplied by 0.2, it is 0.14. P2 and Q1, it is 0.24. Which one is the smaller? This one, P1, Q2. So 0.04 is divided by 0.14, it is 0.286. Now I don't know the logic behind the smaller of these two, why we utilize smaller of these two. I exactly don't know, I have to work out. So this is the normalized D value, 0.286. So rho D value may vary from minus 0.25 to plus 0.25, but normalized D value will vary. Yeah, it ranges beyond that value. Now we can calculate R square, which is D square divided by frequencies of all alleles, 4 alleles under consideration. R square is equal to 0.04 rho D value. Remember that in R square, for calculation of R square, we utilize rho D value, not the normalized one. The 0.04 square divided by multiplication of all these frequencies, it is 0.048. So this is how D, LD values and R square values are calculated. Now to understand the difference between LD and R, I have one example. Let us start with the common ancestor. For one population, we are studying LD in this population. So this common ancestor allele is mutated. There are different points of time where mutation occurred, and in this common ancestor population diverged into many germplasm lines, many genotypes. So you can see here many mutations have occurred, and the single line indicates one haplotype. So this is one haplotype carrying these 3 alleles. There is another haplotype carrying these 3 alleles. Another haplotype carrying only single allele, like that. And there are total 24 individuals present in this population. Now let us work out D and R square for these alleles. So first we consider the green allele. I would call this as yellow allele. So yellow and green are considered together. Let us work out the LD for these two. So here the frequency of this allele, these are the 2 loci. This is one locus, another locus. At this locus, this is the allele, there is counter allele present here. Similarly, at this locus, there is a green allele, its counter allele is present here. So 2 loci and 4 alleles are considered at a time. Now, the frequency P of this yellow allele is equal to 10 individuals where we find yellow allele divided by 24, total number of individuals. So that frequency is 0.42. Similarly, for green allele, independent frequency is 10 divided by 24 because wherever green is present, yellow is also present. So their independent frequencies are 0.42, 0.42. Now their joint occurrence. 5 मिनट में joint frequency जो है, so we can calculate joint frequency considering the individuals where this yellow and green are present together. So that would be the joint frequency. So it is 10 divided by 24. The joint frequency is 0.42. Now we multiply their independent frequency. 0.42 multiplied by 0.42, it is 0.17. Now let us apply the formula of D. D is equal to joint frequency minus multiplication of independent frequencies. That means D is equal to 0.42 minus 0.17. It is 0.24. So this is the raw D value we can calculate. Remember, it is not beyond minus 0.25 and plus 2. It is within that range. Now we calculate normalized D value is equal to raw D value divided by minimum of these two. So frequency of yellow allele is 0.42. Frequency of its counter allele here, this allele, which is not colored, is 0.58. Similarly, here also, frequency of green allele is 0.42. Its counter allele is 0.58. If you work out this, normalized D value would be 1. Now, the range for normalized D value, D dash, is -1 to 1. And surprisingly, this range is also the range for correlation. So D is dependent upon association. That's why it is defined as non-random association between the loci. Now R square is equal to rho D square. Rho D value is 0.24 divided by all the 4 alleles. 1, 2, single locus, another locus, third allele and fourth allele. So R square value is also 1. So there is a perfect linkage disequilibrium considering D dash and R square value. That means these two alleles, R square value also gives us idea about historical occurrence. These two alleles might have arose before divergence of this population. Because they arose before the divergence, this yellow and green, you can see here they are present together in all these individuals. Now let us consider other two alleles to understand the meaning of R square. Other two alleles are this red allele. Here it is present in 4 individual out of 24. So its frequency is 0.17. And I would call it pista allele. I don't know whether the color matches to the pistachio nut or not, but let us call it pista allele. It is present in 7 out of 24 individuals. So that's the frequency of that allele is 0.29. So if frequency of this particular allele is 0.17, the frequency of its counter allele present here in this haplotype, it would be 1 minus 0.17. So it is 0.83. And here the frequency of counter allele for this pista allele, this is the counter allele present. The frequency of counter allele would be 1 minus 0.29. That means 0.71, which is not shown here. Somewhere it is shown here. So just I'm giving you the idea. Now the joint frequency. In how many individuals red and pista both are present? 4 individuals. Both are present. So 4 divided by 24, it is 0.17. Now the multiplication of the independent occurrence, 0.17 multiplied by 0.29 is 0.05. D can be calculated as the joint frequency, 0.17 minus multiplication of the independent frequency, 0.05. So rho D value is 0.12. It is positive. So we'll calculate normalized D value by dividing the rho D value with minimum of these two. When you do that, again you get the value D dash as 1. So you can see here, when you normalize, you get the perfect idea. Here, the rho D value is 0.24. Here, the rho D value is 0.12. But when you normalize, it becomes 1. That's the meaning of normalize. And it is better to compare normalized D values rather than rho D value. R square can be calculated at D square divided by multiplication of all these frequencies. It is 01, square of this divided by all this frequency, it is 0.49. So you see, d dash value, normalized d values are 1 for both these examples, this and these two values. But r square is 0.49 as compared to 1. So from r square, we get the idea that one of the alleles under consideration, it might be the recent evolution. It might be evolved recently. It does not evolve since long time. And you can see here, this pista allele arose before this divergence, roughly time corresponding to these two alleles, but red allele arose recently. So it also gives us idea about the history of those mutations. Finally, the last slide, the difference between LD mapping and association mapping. So what I discussed was LD, and this LD mapping can be performed across the genome. I only explained 2 loci at a time, but we can perform LD mapping of various loci across the chromosome, across the genome at a time. So when you work out the LD between 2 loci, this LD mapping, and when you associate those LD map data with any particular phenotype, it becomes association mapping. That means in association mapping, we are associating LD data, LD map data with morphological trait. That becomes association mapping. So this is a slight difference between LD mapping and association mapping, but in some literature, you may also find LD mapping is used as a substitute of association mapping, but it is not so. LD mapping is slightly different from association mapping. Association mapping is based on LD. When you associate LD map data with phenotype, it's association mapping. So that's all for today. I will continue tomorrow with difference between linkage mapping and association mapping. So that's all for today. If you have any question, then we can consider those questions. Why normalization is necessary? For association mapping, when you utilize germplasm panels, from wherever you have collected the germplasm, the allelic frequencies present in one germplasm line may not be equal to another germplasm line. So there are unequal frequencies present in one germplasm line with another. Now, and because of that, the raw D value calculated is not accurate. So we normalize the D value and we calculate D dash value over the 4 alleles. Now, this normalization is very much similar to the concept we utilize in genetic advance. If you have studied in quantitative genetics, the genetic advance, we have one genetic advance and we have another value, genetic advance as percent mean. So why we calculate genetic advance as percent mean? So that when you divide genetic advance value with mean values, you are normalizing genetic advance as compared to mean of that trait. So the unit will go away. Genetic advance will have raw genetic advance will have one unit. Mean of the trait will have also one unit. Let us say it is plant height, then it is in centimeters. So centimeters are cancelled. We are normalizing genetic advance over mean, so that you can compare genetic advance of one trait, that is plant height, with, let us say, days to flowering on other normalized genetic advance values of one. So once you have the normalized d value, you can compare these normalized d values of any two loci with any other two loci. That's how the LD mapping is possible across the genome. So that's why we are normalizing, because the d value, the raw d value calculated, it is applicable to two loci only. But if you want to compare the d value for two loci with other two loci or any other third loci, then you have to normalize this d value, because the d value is affected by the allelic frequencies. And that's why normalization is necessary, because you might have collected one germplasm line from India, another germplasm line from, let us say, Philippines or America, Europe, and that's how the allelic frequencies are different from differs from one germplasm to another germplasm line. And sometimes these unequal frequencies may create false LD, false association. So to avoid that, we normalize the value of D. That's why value of D is normalized, so that you can compare D value of any two loci with any other loci, other values. So that's how the LD mapping is possible across the genome. That's why this normalization is necessary. So we are just removing the bias from a raw D value. By normalizing it, we are removing the bias caused by the unequal allelic frequencies present in different lines. We are removing that bias. That's why we are normalizing it. This kind of normalization is also done with transcriptome data also. Normalization is a common method in statistics. But yes, the question was valid, that why we normalize the D value. I'm not that sure, but the transformation we apply in statistics may be one kind of a normalization. The logarithmic transformation, arc sine, and square root. Okay, so if there are no questions, then I end our session here. Still, if you have any question, you can write it down. Tomorrow we can discuss that. Thank you. And again, tomorrow is holiday, Sunday, so tomorrow we'll take a break. On Monday, again from 11 to 12, we will meet. So thank you all.